I'm Brooke Anderson, and I'm from the Murdoch-Sunberg FFA chapter. I'm a candidate for Star and Agri Science. In my 2018 project, I partnered with Lauren Krieger, and we tested the nutrient qualities of 3 different types of manure: cattle, swine, and chickens. And for the tests, we used a variety of different test kits. 2 of them we actually have here. Basically, we've had to put the manure in vials, and some of the vials you had to shake, like these ones. We had to put little tablets in there, and then you had to shake it. Then you had to let it sit for a set amount of time, and then you had to compare it to the colors. And sometimes the colors didn't really match up, like you can tell right here. Those are the colors. Sometimes the colors didn't really match up, so our tests actually ended up coming out inconclusive. They weren't really scientific. They weren't the most scientific test kits in the whole world. Right up here is our first one. It was the Accu nutrient test kit. It showed that all 3 types of manures right here was the nitrogen. They were all at the lowest level. This is the phosphorus, and the swine is this orange one, and the cattle is the blue one. They were at the lowest or the highest levels, while the chicken was at the lowest. And then the potassium, they were all the same. So that was kind of interesting. This one's the environmental concept soil test kit. The test showed that the nitrogen in the chicken manure was sufficient, and then the swine manure showed that it was at a surplus, so it was kind of really high. Then the chicken phosphorus showed a surplus as well. Both the chicken and the swine manure had a surplus of potassium, while the cattle manure was just at a sufficient level. The Mate soil test kit, we actually couldn't get readings very well off of that one for any of the manure types, but the phosphorus levels for the cattle and swine manure was at a 0.9 or 9.07, and the chicken manure was at a 3.63. In this bottom one, it's the pH levels. So the pH level for the cattle manure averaged 6.5, while the swine manure averaged a 6.75, and the chicken manure actually was the highest for that one, averaging a 7.75. The Accu test strips were either the same or higher than 0.5. The other trends that we identified in this one is that the potassium levels were the highest of all the nutrients in all 3 for every single test that we did. So after our project in our 8th grade year, after seeing how inconclusive our results were, I actually applied for a grant through the National FFA for $1,000, and I ended up receiving it. So with that grant, we used the money to get scientific testing done on the manure. So in this project, we decided that we were going to focus on just one species, so we decided that we were going to test the feeder cattle. We actually tested 3 different ages. We used 150-pound calves, 500 to 600-pound calves, and then finishing steers. So we actually went through Egg Vice for this one. They sent us containers that were 200 milliliters. So we went out there and collected the manure, like you can see in this picture, and then we cleaned them off and froze the vials before we could send them back to the lab. And after we froze them and got them to the lab, within a couple weeks they had our results back. And those were actually really accurate, and they were pretty easy to read. We were a little bit scared of if we were going to be able to read the results, but they actually came out really well. And in the end, it showed that the 150-pound calves actually had the most amount of nutrients in the manure. And after kind of looking into that, we figured that they would have more nitrogen in the manure because the rumen hasn't fully developed in the calves yet, so they can use a lot, or they don't use as much of the stuff. Then they have a higher protein diet, so a lot more of the nutrients, they actually go back into their waste. Graphs up here, this is the finishing steer 600-pound calves and 150-pound calves. So in this first graph, the total nitrogen levels, so we tested, there's 3 different vials or 3 different containers for everyone. So like finishing steers 1, 1, 1, 2, 1, 3, and then the average. So the total nitrogen in them was pretty close relatively throughout all the trials. When comparing, we had a standard deviation of 0.023 for the total nitrogen, 0.05 for the phosphate and 0.01 for the potash, which was all pretty relatively close. Then when you look at the 600-pound calves, they had a standard deviation of 0.075 for total nitrogen, 0.57 for phosphate, and 0.27 for the potash. And then for the 150-pound calves, the nitrogen had a standard deviation of 0.097, phosphate was 0.05, and potash was 0.17. Since the potash was statistically the same throughout all of our graphs, we just focused on the amount of nitrogen and phosphate in their manure. The finishing steer samples averaged 11.67 pounds per ton of nitrogen and 5.4 pounds per ton of phosphate. The 600-pound steer samples averaged 15.3 pounds of nitrogen per ton and 11.3 pounds of phosphate per ton, and the 150-pound calves was an average of 17 pounds per ton of nitrogen and 6.9 pounds per ton of phosphate. So my 10th grade year, I partnered with Lauren again. We were in a food chem class, so we kind of got interested in that. So we switched from doing plant systems into food systems. And in this project, we tested to see the pH levels to see if they would change after we cooked them in 6 different types of fruits and vegetables. Our tests in this one were actually inconclusive as well. We never really got clear readings on any of them. Again, we used something like that, actually this exact test. So you took these little orange things and you stuck it in the food, and then you had to read it off of the colors again. We thought that when you heated the food, the pH would drop, and it never did. Like, we never got clear readings on any of it. So our tests were neither correct or incorrect because of the lack of consistency. Neither of the test strips ever gave clear readings, but we did have 2 correlations in our tests. The vitamin C for the raw kiwi went from 0.01 to 0.05 in all 3 trials. The other correlation was in the Sunny D and sweet peppers. They stayed at 1% in the vitamin C in all 3 of the trials as well. So for this project, I partnered with Morgan Pearson, and I realized that food products was definitely not my system. So I changed it back, and I actually did animal systems. I tested the efficiency of estrus synchronization in a group of ewes. So some of them are here. We put cedars in to 20 ewes and split them into 3 different groups, and then a week later, we took the cedars out and gave them a shot, a PG600, and put them in with the rams, and then waited for them to lamb, basically. Our hypothesis was proven correct based on our tests. It showed that when the cedars were removed, 16 of the ewes came into heat within 24 hours, and then the other 4 ewes came into heat within 48 hours, giving us clarity. With our results, we can make a conclusion that if you use the cedar implants, you will most likely have a pretty synchronized estrus cycle. When it came to the lambing, we had 10 of the ewes, out of the 10 ewes that lambed within that first week, 19 of them, there's 19 lambs that came. The other 6 lambed 2 weeks later. So there's 19 lambs total out of that first 10 ewes that actually had synchronized cycles. My current project is still ongoing. I partnered up with Morgan again, and we're testing the feed efficiency between 2 different types of show feed to see which animal gains the most amount of weight. The animal systems is definitely the path that I want to go on, and that actually helped me get clarity to what I want to do with my future. I plan on attending Ridgewater next year for a degree in egg business with an emphasis in livestock. And after I'm done with college, I will hopefully go to be a feed salesman somewhere. That's my plan anyway.